Sir, the slides are visible?
Yes, ma'am. Yes, ma'am. It is visible. No problem.
Shall I start, sir?
Yeah, yeah. Sure, ma'am. You can start.
Yes, sir. Good morning to everyone. I'm Shobha, Associate Professor in Jagan Institute of Pharmaceutical Sciences. It is very much privilege for me to give my webinar in my home institution, that is Bapatla College of Pharmacy. I'm always go to the college. Coming to the topic today, with respect to student oriented and also with respect to research, I have decided to share whatever I learned in my PhD that would be going to helpful for the students. The topic is advanced applications of IR spectroscopy in pharmaceutical research and drug development. Not only IR, whatever the instruments we are going to use, how we need to use those instruments for the research, mainly with respect to structural elucidation as well as the identification of the structure. In case of all the fields of pharmaceutical like disciplines like pharmaceutical analysis, chemistry, physics, we may go for a characterization technique. For that techniques, we are going to use these instruments. Specifically, why I am stressing on the IR means, whatever the department you are from, it may be from physics, it may be from analysis, it may be from cognizy. Ultimately, you need to go for at least for the characterization what the compounds are composed of functional groups. So IR is specifically for the knowing the functional groups of the organic compounds. So why we need to do analysis of the compounds means, first we need to provide whatever the extraction you've done. You may go for the synthetic analysis, you may go for the formulation. We need to see there may be any if there be reaction is happened or the synthesis, whatever the happened, it may be impurities having. We are going to see thoroughly by means of IR, especially we are going to see the functional groups. What are those are? It may be have any compound, natural origin like mineral source or marine source. It may be from the plant source or animal source. Every compound is having unique structure. It definitely have some functional groups, especially like carboxylic acids. It maybe have ester, alcohol, or amine or amide. So likewise, we know the at least the basic moiety we need to know. Then only we go for the next step. For that reason, we need to do the structural analysis. It provides the proof of concept, and also we are going to identify the nature. By seeing the structure only, we come to know how many hydrogens are there, how many carbons are there, what are the acceptors or donors are there. Based on that only, we come to know this drug is having this hydrophobicity or lipophilicity likewise. And also we need to go for analysis of chemical as well as the biodegradation studies. Here, some of the scientists, they contributed for the structural elucidation by using X-ray crystallography. She is none other than Dorothy Hartkin. She got the Nobel Prize in the year 1964 for the structural elucidation of vitamin B12. We know that vitamin B12 is very much important for us uh, to treat anemia, as well as the one of the serendipity drug. Serendipity means what? The accidental drug discovery, whatever the foundations led for the penicillin by Alexander Fleming. The structure also, the structure will be illustrated by Dorothy Harkin. For that reason only, for two contributions, she got a Nobel Prize. And also the complete analysis, not only IR, how we are going to do the structural characterization of a compound, where we need to start. Suppose if you have structure or compound, or you are extracted, you are prepared something. How we need to start with the structural elucidation. First, we are going to see with the mass spectrometry, where we are going to get the elemental analysis. How many carbons are there, hydrogens are there, nitrogen are there. Based on that, we come to know the molecular weight also. From that, we have some clear idea, but not completely. Then we need to go for the IR spectroscopy, where we are going to see the functional group analysis. This is a fingerprint analysis, and also we need to go for the NMR spectroscopy. NMR means nuclear magnetic resonance, where we are going to see the relationship between the carbon as well as the proton. So in case of 1D NMR spectroscopy, we are going to see with the proton, carbon, and also depth. In case of 2D NMR, we may go for the analysis of the relationship between the 1 carbon, 2 carbon J coupling. Especially, we are going to use HMBC, HSQC, OC. And also, finally, the important one is X-ray crystallography, where we are going to completely design the lattice of the structure by means of X-ray crystallography. So start with the mass, and also we are going to end with the mass. That is the only thing what we are going to generally perform. With respect to students, they may stop at IR itself only. Only they are going to extract, then they are going for the analysis by means of IR. But not only we are confined to the IR, we may go for other utilization of the advanced instruments, how they are going to helpful for us to know the structure completely. So mainly here for the analysis of spectroscopic techniques, that means by seeing the first slide itself, I have included the electromagnetic radiation, how it will be interact with the matter. Matter means any compound, they may have protons, they may have whatever the electron, that is the only the thing which will lead the show. So coming to the IR means infrared spectrum, where we are going to see the bond vibrations, how the molecules are connected with each other. They maintain some vibrations. Those only will represent in the IR spectroscopy. So mainly here, what we are seeing means mainly whatever the spectroscopy you are going to deal, maybe a spectrometry also. We are measuring the interactions between the matter as well as the electromagnetic radiation. So it may be electronic vibrations or vibrational as well as the rotational as well as spin. The vibrations, whatever it is having, that only will represent in the form of dipole moment. IR spectroscopy only meant for the functional group. No? So functional groups, everything have some more dipole moment. Those only will recognizable in the IR. Without knowing that, we may not see the spectrum for the compounds which are not having the dipole moment. So here we are going to see with the help of IR, we may go for the qualitative analysis as well as the quantitative. Before starting our IR spectroscopy, there may be many contributions from all the disciplines like bioinformatics, chemoinformatics, as well as the laws which are abide by the physics. We already know that quantum mechanics. In case whatever the Schrodinger equation, that will be important for us to know how the atoms are having these energy states. Those only we are going to see by means of these quantum mechanics. That is excitability, how the proton will excite by absorbing the radiation. And also how we are going to see all these things. These are the small equations, how they are helpful for us to know the vibrations caused in the molecule. They may be a simple wave. The wave function will directly gives us the energy states of the molecules. So this is the history behind the IR, not only IR, whatever the spectroscopy that deals with the electromagnetic radiation with respect to quantum. So this is the principle where we developed that the concept of electron, how they are revolved around the nucleus and all given in this picture. And also important equation, Erwin Schrodinger developed this wave function equation. From that also we come to know what are the excitability of the photons present in the molecule. See here, these are the mathematical equations, how they are helpful for us to know that the energy transitions from one level. That means from ground state to the excited state. And also two more scientists, they also contributed for development of this understanding the concept of nature of light. This is the equation, Planck's equation, how it will be helpful for us to know the structure of compound. These are the small theories behind the development of all this structural relation by spectroscopy. So mainly how they are going to be like 3 points. First, whatever the electromagnetic radiation we are providing, that supplies energy to the quantum body. If when it is required, means quantum body has association with the fluctuating electric and magnetic component, and also in the same phase, the applied electromagnetic radiation we are going to give in the form of UV, we may get in the form of visible rays, we may go for the IR, we may go for the electromagnetic radiation. That is the ultimately they will absorb. that whatever the compound that absorbs the electromagnetic radiation, it won't be sit idle. It get excited based on their electronic state from ground state to the excited state. So this is the spectrum of all molecular effects, how they are going to be there. Coming to the IR spectroscopy, it is present in between visible as well as the microwave. So this is the region where we are going to see this IR spectroscopy, how the molecular vibrations are going to happen from downward to the longer length. And also we, apart from the IR spectroscopy, normally how we are going to know the concentration of the molecule by means of Beer-Lambert star. So the observance is directly proportional to the, directly tells us how the concentration of the analyte present in the sample. So overallly for this demonstration, first we need to know the what is molecule, how it is going to have energies at different higher levels, and also how the only the thing is quantum energy bodies, how they are giving and transfers their energy from one state to the another state, mainly with respect to the photons. That is the only thing. Qualitative changes can be induced in the energy level of the molecule by changing the frequency of the electromagnetic radiation. In terms of quantitative, we need to see the changes which are observed by increasing the number of molecules in the path length. That means the length of the sample QET or whatever the sample we are going to make. In case of IR, the sample preparation is like different methods. We generally using KBR pellet method. So all these spectroscopic techniques we are going to use for the structural elucidation of the compound, ultraviolet and visible spectroscopy, infrared, as well as the NMR and mass spectrometry. Raman and atomic absorption spectroscopy is also there for giving the vibrational. So today our main topic is infrared spectroscopy that I'm going to deal in detail. So mainly for the IR active, the compounds need to be have functional groups, and also they need to have some vibrations. That vibrations is nothing but dipole moment. Because of the dipole moment only, the energy will be shared. So these are the vibrations what we are going to express in terms of molecular vibrations. Symmetrical stretch, this is just general theory, what happening, the molecules, how they are organized. In case of symmetric, the bond, whatever the bond length is equal. In case of asymmetric, one will be forward and another with the unequal length. In case of bending, we are having rocking and scissoring as well as the outplane and inplane vibrations. This is the three-dimensional structure of the compound, how they oscillate in a motion. So in case of stretching, we are having symmetrical stretch as well as the asymmetrical stretch, what we have done earlier. And also in case of bending vibrations, we are seeing how the atoms are moving in plane. The bond angle is the same or not. In case of bending, again, we are having in plane as well as the out plane. In case of in plane, we are having scissoring and rocking. Out plane bending, we are having wagging and twisting. See here how the atoms will be moved from the ground state to the vibrational state when they absorb the electromagnetic radiation in the form of IR. That is nothing but Hooke's law. So coming to the Hooke's law, we're going to see the stretching frequency of masses of spring, how we are going to stretch a molecule. From that only IR will be evolved. We need to imagine like every molecule having atoms, how they are connected with the bonds. That bonds only play crucial role for the identification of the compounds. We are having single bond alkanes, double bond alkanes, as well as the triple bond, not only with respect to functional groups like alcohols, acids and all. We may go for the seeing the stretching between the alkenes like a single bond as well as the double bond. So finally, IR is helpful for us to know the function groups. So these are 1 minute.
Ma Shobha,
Yes, sir. Just made, sir.
hello. Okay.
Sir, my screen is visible.
Yeah, yeah. Okay, Ma. Okay.
Yes, sir. Yes, sir. Sorry, sir.
No problem.
So by seeing the electromagnetic radiation, how the all the spectroscopy like ESR as well as NMR, our IR spectroscopy lies in between the visible radiation as well as the microwave radiation. The energy levels are given clearly here, how the electromagnetic radiation interacting with the matter, how they are going to help us to know the excitation stage of the compounds. So in case of IR, we are having near IR, middle IR, and far IR. In case of middle IR only, we are going to see the function group analysis. So these are the instruments we are going to use for the IR spectroscopy. Conventionally, we are following this instrument. Latestly, we are going to see with the IR with microscopy also. And also one more thing, we are having attenuated total reflectance IR spectroscopy, where we are not supposed to do any sample preparation. We are directly going to place a sample under the probe. There we are going to see the spectrum. So this is the instrumentation, how IR is connecting, the source of IR radiation, how the monochromators are helpful to convert the polychromatic light into the monochromatic light that will be fall on the detectors. Most preferably in case of IR, we are using the thermocouple detectors that will help us to know the wave number of that means the distance in between waves is converted into frequency. Here, the instrumentation of the IR, again, I have clearly mentioned how the IR source. These monochromators having rapidly rotating sector as well as one movable and another one is the unmovable. It's statically fixed there. It allows the specific passes of the IR radiation that will be fall on the sample then to the detector. So sample preparation previously NaCl by using the sodium chloride they are preparing the pellets, but is having some disadvantages. Why? Because NaCl is very much brittle and also it is the time taking process to form a pellet. Now it is replaced with the KBr, potassium bromide, which is very much inert and also it is helpful for the sample to much more interaction with the electromagnetic radiation. In case of any solid samples are there, we are going to see with the ATIR. We are going to place the sample we are going to dissolve the sample in appropriate solvents like acetone, methanol, as well as the ethyl acetate. Conventionally, we are using the potassium bromide pellet for the analysis. Here is how the spectrum will be look like. In x-axis, we are going to see the frequency of the compound. In case of y-axis, we are going to see the percentage or intensity of the transmittance. The spectrum will be look like. From this onwards, you need to thoroughly see how we are going to structurally elucidate the compounds by seeing the different zones which are observed in the IR spectroscopy. Whatever the theory we learned, like how it is going to helpful, what the instrumentation, how it is having the all things. Here we are going to practically see the how we are going. That means how we are going to illustrate the structure. See here, some stretching vibrations are there here in the region of 2900 to 3000. We may come to know there may be a possibility of the aldehyde or in ketone. How we are going to blindly tell? So there may be some pattern for remembering. The region rise from the 400 to 4000 region. 400 to 1500, we are telling that is a for metallic as well as the inert compound. From the 1500 cm inwards to the 4000, we are telling that this is a functional group region where we are going to see the absorption or anything, the spectrum comes in this region only. So finally, IR is suitable for the identification and also for the quantification. Mainly, I told you that for IR, definitely the compound need to have dipole moment, then only it will be show spectrum. Otherwise, it won't show anything. There is no stretching or bending. There is no spectrum for those compounds. So mainly, how much the magnitude of the electronegativity difference that in multiplies with the bond length, that is the only the thing happening in the IR. If bond dipole is increased, bond length is increased, dipole moment also increased. If dipole moment is increased, automatically the absorption of the IR rays will be happening. So whenever we want to see the peaks in the compound that need to have polar bond, that need to have dipole moment, then only we will get strong peak. In case of symmetrical, there is no chance of the dipole moment. They will be like, they may be present, but little absorption, or they may be absent. These are the example we have seen. This is acetone structure. Acetone means we know that this is the carbonyl compound where we are going to see the C double bond O. Again, we are having CH3, CH3. For that reason, we'll see. And this is one more. Here we are having the steric enhanced methyl groups around the C double bond C carbon. So there is no chance of dipole movement. These compounds, that is the theory we need to know priorly going for the analysis by means of IR. Here there may be chances of the H is there, no? So maybe some polarization will occur here. For that, we may expect peak, but it is not that much intense than the acetone. These three examples will give us idea. The compound which is need to be get higher peak means that need to have dipole moment. Carefully, we need to see each and every peak and where the wave number it gives, that will give us idea about the functional groups or the stretching which is present in the single bond as well as the double bond. So for student orientation, for GPAT and all, you need to thoroughly prepare in this way. This will be helpful for you to remember the where the functional groups exactly will come at which frequency. See here, I have decided 5 zones for easy understanding. In case of zone 1, it is ranges from 3700 to 3200 cm inverse. In this region, whatever the spectrum we have seen, no? Okay, I will tell you. See here, in case of zone 1, what are the groups we are going to see means alcoholics OH. That means methylic OH we are going to see in the region of 3,650 to 3,200, usually broad as well as the sharp peak we will get in this region. By seeing the peak only, we are going to tell that this having the alcoholic methanol like with alcohol and all. Alkyne stretch, the H is attached to the alkyne group that will be observed in the region of 3,340 to 3,250 cm inverse. Which also having dipole movement, right? For that reason, it also strong as well as sharp peak we're going to see. Some practice problems also there after this slide. Just to concentrate in the zone 1, just we are having alcohol as well as the H is attached with the alkyne, as well as the we are going to see with the amide. Amide means C double bond for NH2. So this the carbonyl group will comes in the zone 4, and amine means primary amine or NH2. We are going to see in the region of 3,500 to 3,300. So only in the region 1, we are going to see that alcohol as well as the alkyne, which is having H, as well as the amine and amide. With 2300 it ends. And zone 2, what are the functional groups we are going to see means, here we are having aryl. Aryl means benzene, the aromatic compounds which obeys the Haeckel rule, as well as the vinyl, sp2ch. And also in case of this one, we are going to see alkyls sp3 hybridized carbon as well as the aldehyde. So aldehyde, amide, and carboxylic acid, ester, and ketone. Those are having two functional groups. That means they are appear in two regions. One is in the fourth region, and another one in the second and first. Why? Because they are carbonyl compounds. They are having strong electronegative atom, that is oxygen. So they have more polar nature. For that reason, it having sharp and intense peak at the region of 1700. So zone 1 is completed, where we are going to see the alcohol. The H is attached with the alkyne as well as the amine and amide. In the zone 2, we are going to see the aryl vinyl sp2ch. Not completely the say double bond sp2ch, and also here the 2,900 to this 2,800 region, we are going to see the sp3. That means alkene stretch. And also the sharp, we need to gunshot it. We need to remember this thing in the region of aldehyde, that is sharp for the aldehyde groups 2,900 to 2,700. Whenever we saw the peak in the region of this region, we come to know the compounds may have the aldehyde group. And also carboxylic acid, where we are going to see in the region of 3,000 to 2,500. The peak's shape is different. Here we are going to see the broad. For these things, some may be short, some may be broad, some may be shorter. Especially in case of amines, we are going to see the smaller peak. And also next, the zone, whatever the zone we are telling now, that will be like only we are studying here, the function group region only, where it starts from the 1,500 to 4,000 cm inverse. So first zone is completed, 3,700 to 3,200, and second zone is completed in between 3,200 to 2,700. And also third zone, that is lies in between 2,300 to 2,000 cm inverse. Especially for third zone, it will be like a triple bond. Likewise, also we can easily remember where we are going to see the alkyne triple bond as well as the nitrile, that means cyanide stretch in the region of zone 3. Here also the peaks are maybe sharp and also variable. Size of the peak is not broad, maybe vary according to the groups which are adjacent to these compounds. So zone 3, triple bond, likewise you can remember easily, alkyne as well as the nitrile. And also the important region where we are going to see this carbonyl group stretching means, in case of carbonyl compounds, we are having aldehyde ketone, and also these say ester as well as the carboxylic acid C double bond OH, and amide C double bond NH2. We are going to see in the form of this zone 4. So already I told you carboxyl group will be seen in the zone 2 also. The C double bond O will come in the zone 4. So whenever we want to see the structure, they having the carboxyl group, we need to see the both the regions. In the zone 4, we need to expect C double bond O, and in the zone 2, we need to see the broad peak which is having the appearance of the peak in between 3000 to 2500. So for amide also, we are having the peak at 1690 to 1650 cm inverse. Frequency we are representing in the form of wave number. That's why we are giving this unit centimeter as E inverse. And also last June, for the remembrance, these are like a benzene containing compounds. They need to have C double bond C here in the region of 1600 to 1450. Whatever the C double bond C in the benzene, it comes in the second June here. For benzene also, we need to see double bond C will come at 5th June and 2nd June. It might be confusing now, but no need to worry. You need to practice. That will be helpful for you in learning. So here in Zone 5, we are seeing the alkene as well as the benzene stretch, especially double bond, C double bond, C, the alternative double in benzene means what we are having C6, H6, alternate double bond means resonance it is having. So when we are thorough with the IR interpretation of the compounds means we need to have thorough knowledge as well as the thorough practicing. So if students are available, sir, they need to note these compounds. Systematically, how we are going to give the nomenclature, that means functional group analysis in the IR. By seeing the structure, you are seeing here CH2, 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 it means alkane. Here is the alkyne structure. So I told you whatever the H that is attached to the alkyne, that will be comes in the zone 1. Zone 1 means here 3700 to 3200. So clearly I will give you idea about how the peaks will be appear for this compound. Especially this is like a aliphatic compound where we are going to see the alkane chain. At this adjacent, we are having the alkyne stretch. So first zone, we are going to see this sharp peak, that is H, which is attached to the alkyne. And also here, the 3000 second zone, we are going to see the sp3 hybridized carbon. And also I told you third zone, it lies in between 2300 to 2000, where we are going to see the triple bond, that means valency 3 containing compounds, especially alkynes as well as the cyanide, that is nothing but nitrile group. So this ends with this compound analysis. How easy it is for us to know the zone analysis. So here by seeing the scale 500 to 1500, we are telling as a fingerprint region where we are going to analyze the metallic compounds as well as the inorganic compounds. Inorganic means other than the carbon. Here we are going to clearly seeing that the functional groups will be helpful for us from 1500 to 4000 cm inverse. There may be some overtones of the CC single bond stretchings here. Another example is here. Please look into this. It is having double bond as well as the aliphatic chain. So by means of understanding, if anybody is really following, they are in the position to tell me. It's quite difficult for me also to interact with you people. Okay. By seeing the structure, we come to know which is having aliphatic alkane structure as well as the C double bond C. By analyzing systematically from zone 1 to see here one by one. So whatever the CH2 that is having with a C double bond group, that is coming in the zone 2. And also the aliphatic region, we need to have scale in between exactly 3000. We may note that, which is having aliphatic stretch. And also the double bond, whatever we are told, that will be observed in the region zone 5. And also one more example, by seeing the broad peak in the region of zone 1, that is lies in between 3000 to this 3500, especially in the region of 3200, we are seeing this alcohol, that is aliphatic alcohol, methanol pH. Remaining is aliphatic chain. This is broad. Another example with respect to carbonyl compounds. See here, the aliphatic stretch is exactly where we are going to place the scale at 3000. The whatever the stretchings that will be appeared in this region are aliphatic stretching, and we know that the sharp peak, intense peak will be appeared for the carbonyl group, that is C double bond O, in the region of 1700. It may be vary depending on the number of substituents attached to the C double bond group. Very strong peak we are going to see. Just observe the things. So IR spectrum has divided into 5 zones for our easy understanding, where we are going to see these things. Zone 2 as well as a zone 3, 2300 to 2000. Once you need to practice zone 4, where we are going to see the carbonyl compounds, and zone 5, where we are going to see the C double bond C as well as the C double bond C that is present in the benzene. This is the fingerprint ratio. So just to see the things, I have given the work here. Sir, if students are there, please take down the notes, sir. Otherwise, I will continue myself. So by seeing how, first thing what we done, by seeing the structure, we are going to give the zone analysis. Here, the another pattern where we are going to give the chemical formula, molecular formula. From that, we need to derive the where the functional groups will come by using double bond equivalence. So first we need to calculate the double bond equivalence by means of this formula C minus H by 2 plus N by 2 plus 1. We need to substitute the number of carbons as well as the hydrogens as well as nitrogen. In place of nitrogen, we may go for use of halogens also. From this, we come to know how many double bonds are there, whether it is having ring structure or aliphatic structure. So for this compound, what we are having, C6H12O2. So 6 carbons, 12 hydrogens as well as the oxygen 2. By substituting, we are getting 1, means it is having 1 double bond. So we can derive the structure by seeing the peaks which are present in the IR as well as the molecular formula. So we may expect that from this formula, after calculating the double bond equivalence, we may thought that the compound may have pi bond as well as the one ring structure. So one ring is not possible. Why? Because we cannot see anything in the region of 2300 to 2000. That is the impact which is giving from the peaks which are present in the spectra. So carefully assign the structure. Sorry, sir. Correcting, sir. Sir, this
Madam,
is
okay.
yes, sir. Thank you, sir.
Right. Okay.
So upon calculation of double bond equivalence from the molecular formula, we come to know that the compound may have one ring or one pi bond. By seeing the peaks which are observed in the IR spectrum, we made that in the zone of one, we have seen that one broad peak. We thought that we're having OH. And also in the molecular formula, we are seeing the O is there. So clearly, systematically, we need to illustrate the structure by Jones analysis. In case of zone 1, we are seeing alcohol. There may be a possibility. And what we are going to see in the zone 1 means we may expect the peaks for alcohol as well as the amine as well as the H is attached to the alkyne CH. So this by seeing the broad peak, OH is present. There is no short or anything for the amine. And also there is no chances of the triple bond. Why? Because they have by calculating the DBE, we thought that there may be one pi bond. So there is no chance of the triple bond. And also zone 2, we need to see where it lies from 3200 to 2700 cm inverse. In zone 2, we are having some aliphatic stretch. Exactly where we are going to place a scale at 3000 cm inverse. So completely with not all of us, we need to thoroughly and steadily look after these things. So maybe there may be a chances of SP2 means maybe this is the aliphatic stretch. Maybe definitely they may be having some small stretch at the 3000 besides. And also there is no chance of the aldehyde. Why? Because aldehyde, especially for aldehyde, definitely we will get the peak at 2900 to 2700. So there is no peak. We come to know that this structure won't have any aldehyde. And also for carboxylic acid, also there is no chance. Why? Because for carboxylic acid, what we thought means it may give broad peak but shorter. So zone 3, see here in zone 3, there is no well-established peak. So we can avoid this zone 3. There is no possibility of the C triple bond C. We can exclude from our analysis. And also there is no chance of the cyanide group also. Then zone 4, which ranges from the 1850 to 1650. There may be a chance of the carbonyl compound, but we cannot show that. We already excluded the carboxylic acid as well as the amide. There may be a possibility of the ketone. Ester means no enough oxygens or CO1. There is no aldehyde peak and also no carboxyl group. And also for amide also we are not having any nitrogen in the molecular formula. Only we are having C6, H12 as well as the O2. Maybe possibility here by observing the zone 4 analysis, we may have the presence of the ketone. Why? Because from the zone 1 onwards, we are excluding each and every one. In zone 1 only we have seen broad alcoholic pH. In zone 2, we may have aliphatic stretch. That is the only conclusions we drawn from the this analysis. In case of zone 5 also, see completely there is no any benzene stretching. Why? Because there is no satisfaction of the white. In case of benzene, we are having 3 double bonds. So only we are having 1 pi bond means we can exclude this benzene ring from giving structure. And also there is no alkene structure also. So finally, this is the structure of this compound, the actual structure. So we cannot predict the structure by means of this DBE as well as the IR. So how beautifully we have elucidated from the molecular formula by taking these IR peaks. This is the beauty of IR spectroscopy, where we are going to derive the structure from the peaks as well as the molecular formula by applying DBE. So one C double bond OH is there as well as the metallic OH is there, remaining aliphatic stretch. So another example here, likewise only we need to go for the systematic analysis. First, we need to calculate the double bond equivalence by substituting in the formula. After that, we come to know how many rings are there or how many unsaturations are there. From that, we need to see the is there any chances of the presence of these amine likewise. So here also same thing, same formula. C minus H by 2 plus N by 2 plus 1. If students are there, if you are there not in the system to learn, I will share my PPTs. Please practice, do the practice. Amine in the formula, but not that much sharp peak in the zone 1. And also this CH stretch also not there in the zone 1. Next zone 2, where we are going to see this aliphatic as well as the aldehyde. See clearly. There may be some aliphatic stretch. Maybe this sp3 hybridized carbon CH stress is there. Aldehyde may be present, but not that much sharp peak in the region of 2900 to 700. And also we are not seeing, we have even one more thing here, we are not having any sharp peak in the region of 4th zone. And also in the carboxylic acid also, there is maybe a not having sharp and broader peak. So from this zone 3, there may be a chances. Why? Because in case of 2300 to 2000, we are having peak. So by seeing this peak, we need to thought that there may be a possibility of any triple bond containing alkyne or cyanide is there. That means nitrile. See here, it may be a possibility of the alkyne or cyanide. Why? Because the presence of peak itself indicates that indirectly tells us there may be a chances of triple bond containing compounds. In zone 4, there is no sharp intense peak for the carbonyl compound. Zone 5 also, see zone 5. Maybe chances are there. The presence of the C double bond C in the benzene ring. So this is the final structure by means of molecular formula only. Same thing we have substituted in the double bond equivalence formula. Then by seeing the peaks systematically from zone 1 to zone 5, we have drawn this structure. This benzyl having benzyl nitrile structure. So mainly, what are the advantages of IR spectroscopy? The main advantages of the IR spectroscopy is, first, with respect to students, they may go for the analysis of IR. The cost of the sample analysis is also less, and also the sample which is required for the analysis is also less. So that is the only thing we are having mind to go for IR spectroscopy when compared with remaining. And also it is a non-destructive. There is no preparation of the sample will be taken place. And also mainly helpful for the identification of functional groups. We are going to see the in between as well as the within the molecule, how many hydrogen bonding are there. That is nothing but dipole movement. By means of dipole movement only, we are telling the stretching and whatever the vibrations we are seeing in the IR spectrum. And also the chemical interactions also we are going to see. If we want to formulate anything, you want to react to something, there may be a, suppose we are going to take the alcohol and acid. By combining this, we will expect the ester. The ester group will give these significant peaks in the region of this zone 4 and zone 2. Likewise, only we may go for the analysis of individual peaks. From that also, we can tell that whether the interactions are taken place or not. And also it is helpful for us to identify the structures of the solid in the form of solid also. In case of liquids, we may go for the ATIR, attenuated total reflectance, and also gases can be analyzed. This is very much affordable, cost effective method. Mainly with the drawbacks also there for IR, those are nothing but quantitative analysis. Complete quantitative analysis is not possible. And also, if you have any herbal extract, there may be a lot of functional groups in the phytoconstituents that won't be possible with the, not clearly possible with the IR. And also, complete structural isolation not possible. Why? Because we are only giving the functional group analysis only, not the entire structure. For entire structure, we may prefer other spectroscopic techniques like NMR as well as the instruments like other chromatographic techniques like LCMS as well as the GCMS. And also this have some impurities also. Microplastic analysis also we can go further using IR. This instrument, whatever here showing the IR spectrum having the microscopic analysis. Just we are going to play the sample. It will give the microscopic pictures. From the pictures also, we come to know these are the polymers and these are the functional groups it may have. So micro to very little sample is enough for the analysis of IR spectrum. These are the microplastic analysis. Whatever what composed of polyethylene, polypropylene, polyamides, polycarbonate, polyethylene terephthalate, polystyrenes, polyvinyl chloride, cytosine based polymers also we are going to prepare. No? For that reason also we may acquire this IR spectrum for complete the stretchings which we expect in the molecule. We can, and also one more important thing in the IR spectrum, the software whatever we are using, and generally Opus or any software which is linked to the IR. In that software, we are going to give some commands. From that, we can correlate whatever the compound or excipient or anything by we can superimpose at a time. At least up to 10 compounds we can superimpose and we can compare the results whether what is the difference in between and among them. That will be possible with this IR spectrum. From that also we are having clarity about what is the initial product and what is the final product, or in between whatever the things happening. To this I think completed. In case of zone analysis only, please look after these things and learn properly. That will give you more information about the, in case of GPAT also, we can shortly will answer the questions.